Good morning, everybody. Today is our fourth event for this webinar. We do it every month and exchange ideas between the professionals. And for today, we have Nujhat Maisha, and she is currently working as a cell and gene therapy scientist in 2seventy Bio Incorporated, developing autologous cell therapies for oncology treatments. She is part of the process development team working on formulation development in early and late stage manufacturing. She has extensive experience as a bench scientist in designing and characterizing polymeric nanoformulations for targeted drug delivery systems and biomedical applications. Her educational background is a bachelor's in chemical engineering from BUET and PhD in chemical engineering from University of Maryland. We are delighted to have you here today sharing your unique journey from academia to industry. So with that, I'll let Nujhat start the presentation. Thank you.
Thank you so much. Hoping you all are having a good day. First, I just realized I have my date set as 2 days ago, so struggling with keeping up with the days, but so today is 15th, but yeah. So yeah, thanks for giving me the opportunity to talk about my experience over the last few years as I transitioned from academia to industry. I'm currently working as a scientist in process development, and I wanted to highlight today in this talk about my thoughts that are mainly relevant to the biotech perspective and might be more relevant for chemical or biomedical engineers who might be in the same path as I was. And these are also thoughts of someone who has very recently been on this path, like in the initial years of like transitioning to industry. So I am guessing many of you may have more experience in this process of finding the right opportunities. So I would love to hear more your opinions as well. So after getting my bachelor's in chemical engineering, I started graduate school where I was working as a research assistant from 2016 to 2021. And during those years, I was involved in developing polymeric nanomaterials, mainly for drug delivery and biomedical applications. I was also working on developing relevant analytical tools for these. After that, starting from 2021 to present, I've been part of the formulation science process development group of 2seventy Bio, and I've been focused on developing and optimizing viral vector processes from the unit operation, starting from purification to fill finish, and also focused on improving the yield and transduction of efficiency of these viral vectors. I'm also working on integrating existing and novel characterization techniques for better understanding these viral vectors as well. So through this talk, I mainly wanted to go over a short overview of the lessons learned, mainly by trial and error on my part. And in this talk, first, I wanted to go or highlight more about my previous experience developing the polymeric nanoparticles. And I would also try to cover in each section what were the key skills that I had obtained in that process as well, that I believe helped me in making that transition later. So I started working in a group in the University of Maryland, Baltimore County, that was heavily focused on developing biomaterials for drug delivery and biomedical applications. My lab had been working for decades developing a technology which is called hemostatic nanoparticles. It is targeted for trauma, where the nanoparticles, if injected into the bloodstream, it goes into the site of injury, helps in forming a clot faster, and reduces blood loss. So my group had worked extensively with small animal models like rodents and found that it was effective in reducing bleeding time. However, the main challenges or the main obstacle was finding out if these were compatible with large animal models and eventually humans. And that's where they faced hurdles where it actually showed that it might have even led to some infusion reactions. So that is the time where I got involved with this project as well. And one of the main things that we started with was trying to understand the biology behind what was happening in these large animal models after the nanoparticles were injected, and utilize that knowledge to develop our next generation hemostatic nanoparticles. So first, we took a step back to understand what happens when nanomedicine are injected into the bloodstream. We went through available literature and realized we were not alone in this. And similar infusion reactions had been observed for several drug products as well, such as nanoparticles that were lipophilic and used for delivering chemotherapy drugs as well. These infusion reactions can range from mild to moderate, but in some cases, it can also have severe outcomes. So we directed our efforts in developing clinically translatable nanomedicine. And in order to do that, we first worked on developing bioassays that would be highly sensitive, and then utilized those to validate our in vitro response in vivo for novel nanomedicines that did not lead to such infusion reactions in vitro, and then deploy the designed assays for understanding the impact of properties of materials better so that we could develop the next generation of nanoparticles as well. So as a first part, we worked on developing in vitro assays with high sensitivity. So when we say in vitro, we mean that these assays would be able to depict what is supposed to happen inside the body outside using biological fluids such as blood. So this could be critical in trying to even evaluate whether a material is compatible even before we start an animal study. So it gives us the opportunity to tune our materials as best as we can so that it leads to the most safest profile when it is eventually in the bloodstream. So we worked on understanding what are the optimal conditions for evaluation of complement response in vitro. And as we worked on that, we found that we were able to develop an assay with high sensitivity for evaluating complement response. And we found that for many of our in vivo data that we already had for those same particles, our assay was able to mimic the same kind of trend in as well in vitro. That is what we see happening once it is in the animal. We saw that same response when we were testing it in blood on the bench lab as well. So the broader impact is that it gives us the possibility of screening nanomaterials before moving into experiments involving large animal models, or even potentially we could replace large animal models at some point with this tool, which is sensitive enough for screening the nanoparticles. So if I had to summarize what were the skills obtained from this initial part of the research, I would say it would be extensive knowledge about the analytical techniques involved, experience in developing bioassays, and mostly experience in developing assays that were rigorous and stable as well.
Nuzhat,
Sure.
I have a little question.
Yeah.
These are quite technical.
Yeah.
And can you explain a little bit on the bioassay? What is that?
Sure. It's actually an assay that is more, that is mainly for like biological fluids or cells in general. So what we try to see here is that like it would be very similar to any other chemical analytical technique, but I would say we see the response for the biological changes. For example, if we are trying to track, suppose, an allergic reaction, there are some markers that would be high if the reaction takes place and low in others. So these assays are mainly able to predict those, but outside the body. So we use fluids like blood that may have those components that can lead to that kind of response. Does that overall help in explaining that better?
A little bit, yeah. That's good enough, yeah.
Okay.
Thank you. Yeah,
please
you can
do
move
interrupt
on.
me and ask me questions, because I think, I try to make it more of it would be
right?
like more like it's like for all backgrounds, because I think when I try to explain it, I go back to the fact that I'm talking to a room full of like biomedical engineers, but let me know if you have any questions.
Will do. Please
Sure.
go ahead.
For the second part, we worked on now coming back to our nanoparticles itself, like how we can make those nanoparticles better. And for the starting part, we looked into the existing nanoparticles that we were already working with and the surface of those nanoparticles. So what we basically want to do is that
now I'll stop you again. Sorry.
Sure.
These nanoparticles, what are they made of?
These are polymeric nanoparticles. The polymer that we work with is polylactic acid. It's a biodegradable polyester So similar materials are it's FDA accepted and even FDA approved in some cases. So these are materials that had been used extensively for years in this biomedical engineering field for drug delivery applications, for making patches, even the sutures. Like sometimes when we have an injury and we get sutures that are biodegradable, those are made up of polyesters like this, polylactic acid. So like these are like highly biodegradable and compatible for human use as well.
No, that helps. That helps. Thank you.
Sure.
Yeah,
So,
this is some sort of polymer acid, right?
yes.
Okay. I mean, okay. Thank you. Move ahead.
Sure. So we worked on understanding the surface of these nanoparticles. Like basically, we wanted to create these nanoparticles in such a way that it would be in stealth mode. That once it's in the blood, it won't be understood as something that's like not like native to it, rather, and like there would not be an infusion reaction. So that was our goal overall, to make particles that were stealth. So we tried to do that by adding polyethylene glycol on the surface. Like it was already a part of the original nanoparticle, but we tried to understand like if we played with the size of the polyethylene glycol or other properties of it, then could we control it? Now, I would also like to highlight like polyethylene glycol is also FDA approved, and it is highly used for even increasing the circulation of a number of drug products in the bloodstream, because what it does, it helps in leading to higher circulation rate for many of the things that have very short half-lives and could easily be like out of the system. But this just helps in keeping a higher drug availability overall, I would say. So what we found for this was that the polyethylene glycol helped in reducing our infusion reactions, and these were mostly tracked outside the body, that is using our assays with blood only. So we were just mainly measuring that the tracker for infusion reactions, if those are high or low, or if they remained same. So we found that the polymer, the nanoparticles that had highest amount of polyethylene glycol on the surface, that had least amount of those markers present. We also compared different molecular weight of those polyethylene glycol on the surface and different combination. So eventually we came up with a nanoparticle that led to least amount of those responses in outside the animal studies, or in like in the bench scale. So the next part is like, once we had that nanoparticle, if we can now test it in animals to see what's happening and if we are able to get the response that we expected. Now, these are meant to be hemostatic. So we had a porcine trauma model with pigs, and these nanoparticles were injected into the bloodstream. So the first part or takeaway is that these were able to promote clotting. We measured it through prothrombin measurements, which is a measure of the clotting time. We then also tracked the same biomarkers that we tracked in our lab scale and found that we don't see signs of infusion reaction as well. However, this study also led to as we got more data and also looked into more physiological changes, what we see is that we do see signs of clots in uninjured tissue, and that brought us or introduced us to another challenge or hurdle that we tried to solve in the next steps as well. But in summary, if I wanted to summarize the skills that I had obtained from this part, it would be working on the materials properties, developing polymers, understanding the material and polymer properties better, and also characterization of nanoparticles. One thing I also wanted to mention, that we also worked with collaborative teams, because the signs of clots that I mentioned, we were not able to decipher what was going on just on our own based on the data we had. So we got help from data scientists who took the data that we had for different of those components, like IL-6, lymphocytes. These are all components of our blood, and they may increase or decrease based on different things happening in the body. This could be because of inflammation, injury, or different things. So they took that data, tried to apply different statistical methods to that, and then see which markers were showing critical changes. And that was able to give us some direction that in our next generation particles, what do we want to keep track of? So basically, we worked in a very collaborative environment with immunologists, and data scientists from different groups. Now, overall, if I want to summarize what most of my journey in the graduate as a graduate research assistant work was, it was mainly finding that surface coatings can control, but not entirely prevent the infusion reactions. And while it was helpful in reducing interactions, we still needed to rethink what would be the optimum biocompatible material. So most of these works were funded by DoD grants and NIH research grants. And in the next step, we actually tried to look into a number of different materials. And I would keep this brief and just try to highlight the main point, like what we eventually landed on. So after our extensive work with different materials, including surface properties and material nanoparticle cores, we found that we were able to generate a batch of nanoparticles that were stealth. These are more of a core shell kind of structure and made up of polyurethane, and they were able to evade the infusion reactions as well. So we tracked the clotting profile as well, like whether it is able to form the clots as we expect it to. And we did these outside the body using blood and a technique called rotational thromboelastography that can show us like whether clots are forming or not. And we were able to see that these can help reduce the time to form clots. We also compared that what was the change in the biomarkers. Overall, the closer to 1, the closer it is to the original state. That means we are not seeing any change in the biomarker profile. And we found that compared to our previous generation nanoparticle, almost all of the polyurethane nanoparticles were showing much lesser amount of biomarker that could be sign of infusion reactions. So I'll slow down here and ask if you all have any questions so far.
Well, yeah, I do have questions. So from you moved this polyurethane, right? I mean, this is was different from what you started with.
Yes.
And this one is like showing, you know, when you say stealth, and I think of really the stealth fighters, right? I mean, it's very similar concept here that you are injecting something and that's not really disturbing the rest of the bodily fluids and other things and able to do its job, right? So this polyurethane, how, I mean, was it in development or you guys kind of introduced it?
We did some literature readings actually, and a similar kind of material is used for dental resins and many other biomedical applications, but not for injecting it into the blood. So we were the first group to try to see if we could like see what the responses are when it comes in contact with biological fluids like blood. So
okay.
As we took that concept, we then again worked on the surface part. Like the original concept that we started with was just how to make these polyurethane nanoparticles, but then we modified it to add polyethylene glycol on the surface because that helps in improving the circulation. It also is useful for targeting to our like the parts that we want to target it. Polyethylene glycol helps with that as well. So we did some work on that as well. We actually have currently like a patent application for this that is that we have been waiting for more than a year now. So we have been working on the patent as well for this.
When you say like how to make, I mean, I'm sure there's a team of people working here, right?
Yeah.
And how to make, are you involved in that group? Like,
yeah,
I made also say this is how it should be made?
Yeah, I actually made these nanoparticles and
you did?
Yeah, so I made those nanoparticles, characterized them, did the assays. The only part I think so far I've mentioned like the animal studies, those I did not actively take part in. We make those particles, and as I mentioned, we have collaborations with DoD that funded our study as well. So they have like an animal facility with veterinary doctors and everything who can like design those kind of big scale studies. We generate the material and they conduct those animal studies and help like provide us the data back to try to see what's happening.
Okay. So no, that's good because I mean, I'm in the like a manufacturing industry. So I'm trying to find similarities, right?
Yeah.
When you are developing these particles, there is that, how do you, characterize it? What do you use to measure certain characteristics? How repeatable those measurement techniques are, right? I mean, those things interest me, right? I mean, we can go more into it. I mean, I can ask more questions, but as far as the presentation, I mean, are you gonna do some more? I mean, and then we can get into the overall, discussion part of the presentation.
Yeah, I think I'm almost like very close to that part, like how it all helped me in making that move, and also what were the skills that actually helped me. So
okay, let's do that, and then we'll all participate in more discussion.
Sure. So overall, I would say like it had helped me was in designing robust studies utilizing novel materials. And when I say robust studies, it also includes like robust analytical techniques like measuring these particles and making sure we get consistent batch to batch replicability. Also making sure even the assay is like rigorous in the sense that since we use biological fluids, every donor will have a different response. But at the same time, there should be a similar kind of trend that we see. So if we see that kind of similarity in those donor to donor variability with the donor to donor variability as well. So overall, I would say if I had to summarize the outcomes, it would be developing a highly sensitive screening assay and also understanding more about materials properties, which eventually helped us in designing a nanomaterial that is showing those stealth properties that is essential in overcoming infusion reactions. Now, the main question is like, how do I utilize these outcomes into a meaningful skill set for the industry? So currently, as I mentioned, I'm working in developing viral vector formulations for manufacturing autologous cell therapy. Viral vectors are the starting material for the drug product we make, which are the CAR T cells. And we develop bench scale processes that are like much smaller than the actual large scale manufacturing. And after we make those processes, like we have established some like processes bench scale, we then work on transferring those technology for large scale manufacturing, which are always like good manufacturing, like making sure they're like GMP compliant, they have GMP compliance and such other things. So this is just a process flow diagram for what the viral vector production system process looks like. This is taken from a site from Cytiva Life Sciences website. And this process is for adeno-associated viruses, but for all viral vectors, the process is more or less similar, where there's an upstream part where first the viral vectors are produced by infecting particular cells of interest, and then they are harvested in the downstream steps and eventually purified, leading to the final product that is filled into appropriate vials. So the activities that I'm involved with are mainly starting from formulation up to the final product filling and finish. And as we are a part of the development work stream, what we do is that we make sure our formulation is robust.
Hi, how are you? I'm in a meeting actually.
So yeah, we make sure like our formulation is robust by ensuring that at different temperatures, they show the same level of stability. They are able to maintain the ability to be infectious as we expect it to be for making the actual drug product. So this is a generic job opening posting for any formulation scientist role that I picked from Google today. And just to show like what are the main qualifications that are expected for such a role. And as you can see, like it's a very competitive kind of industry in the sense that my peers are not from the same kind of background. It's a very diverse field. So some folks could be from biochemistry or bioengineering or chemical engineering, or even from like other fields of biological sciences. And then the experience is also very different and also very specific from like one person to another. For example, some people might be joining or looking into this role after completing a PhD, whereas there could also be people who are competing for the same role who might have been in the industry for 5+ years. So also, if we look into like what are the skill sets essential, this could be a number of analytical characterization skills, and also ask for skills like communication skills and project management skills like organization, time
It's going
management, and
slow,
such other
bro.
skills.
It's going slow today.
Oh, sorry.
Yesterday it was fucked up. 600 ka.
So
Excuse me,
$6,000.
Mr. Abdul
Sale, yeah.
Mannan, can you please mute
Yesterday
your
it was
phone?
fucked up. Also, if
Uh,
there's a lady, then you are done.
Okay.
Why?
I think
They never
he's not hearing
just
me.
hold like this.
Let's see if I
Two
can
friends of friends. So
Can someone mute him?
mute him. Yeah, that's what I'm trying.
she always hold like this.
Oh.
And that's it. Huh? And
Chat.
always Barun walks around here.
Can you
and he always
please
stay
mute
behind
yourself?
Barun and Barun will are they in relationship or something then? Then? But they weird.
Okay.
I think the
I can
organizer
match with
has
the
to
friendship.
mute.
Yeah.
Organizer should be able to unmute him.
In a meeting. Or in a,
I was also there in a meeting now.
just going on, but I'm not taking everything. When my part will come, I'll take.
Yeah.
Okay, I think I can continue. So as I was mentioning, like what are some of the transferable skills that I had that might have helped? I think one of the main requirement for positions like this in industry is ability to independently design experiments, analyze data, and try to draw like mainly like drive studies that are hypothesis driven, and also utilize previous experience in analytical characterization as well. Another thing I wanted to mention, like the previous slide I showed, like there were a number of analytical skills included. Now, one might not have all the analytical skills. However, previous experience with some fosters the fact that many of these skills can be acquired over time. Also, I guess working with a cross-functional team, the requirement for that is often often covered by the fact that we have that experience of working in collaborative projects with people of different backgrounds and different sectors, like starting from the immunologists to like data scientists and all of the things. So also wanted to highlight that the fact that we are used to failure in academia, that also helps or makes our case stronger, that we are able to adapt and make comeback plans faster as well. So as for like a little bit more about like what are the lessons learned through trial and error, I think it would be the first one would be like how to make a resume that is more catered towards industry compared to academia. Like a general resume for academia is usually much bulkier. It has details about like all the projects that I might have worked on, all the people I had mentored maybe as a part of those graduate programs, talks given, and things other, like all the highlights about the different projects. But as it comes to reformatting that resume, the first part is simplifying it to highlight the aspects that are more relevant for industry, even for a particular job in general, highlighting the skills of what kind of project management skills I had gathered, mentoring experience gathered, making sure that I have some of the keywords that are mentioned in that job opening as well in my resume. That could be analytical skills, that could be requirements for process, like designing experiments and many other things like that. Even skills like statistical background, things like that. And lastly, it's important that resume is also catered for each position applied to. For example, if I'm applying to a role that is for process development, I would mainly highlight my experiences in developing materials, executing and designing those hypothesis-driven studies. And while I do have a good amount of experience in developing assays, that takes a backseat in this resume overall. But if I'm trying to actually apply to a position for analytical sciences team, I think that's where I then redesign it to bring that analytical experience in the front part and making sure I can like highlight that as much as possible. Last one is to learning to reach out, making connections, because that is something that I struggled a bit, because for the biotech industry, there are not a lot of Bangladeshi people that I knew personally. and they're like the number is increasing over the years, but there are not a great lot of Bangladeshi people in that field. So the first group of people that I reached out to were alumnus from my graduate school, and they were actually very helpful in trying to introduce me to the different kinds of roles they were in, what's usually expected from those positions, or even reviewing my resume for things like that. They had been a very helpful community in that part. Also, another part is like making connections with many of those similar kind of like people working on those similar kind of backgrounds. Like for the role, suppose if I'm working or looking for a role in like downstream processing, trying to get connected with more people who are actually in that field and understanding what they are looking for in positions like that. And one thing that I did not have a lot of experience with was exploring co-op opportunities or internship opportunities, but that is always also a good starting point during the summer internship, through like the summer internship programs and things like that. So that is most of the content that I had for today, and I would like to take any questions you might have at this point.
Thank you, Nuzhat. That was the awesome presentation. But I like to mention one thing to you. You bring up a very important point in there. While we are transitioning from academia to industry, and reaching out or networking is very important. And oftentimes, as a minority group in this country, we feel that we don't know anybody like you. If you are working in a non-popular field among the Bangladeshis, that's even difficult. But the point you mentioned that you reach out to the alumni, that's a very good point for everybody. So if you don't know anybody, don't hesitate to reach out the alumni. That's I really.
Yeah, I think I can also add one more thing to that. Like, I started my job search process very late, in the sense like I was, I think, more interested towards academia up to my 4th year of PhD. So it all like started having a snowball effect that I have to make more connection, and there's not enough time to like go to more like networking events. And it was the time of COVID, so we were not having like a lot of face-to-face interactions as well. But there are actually a good number of like events taking place in like most of the graduate schools where employers come in for like those workshops and things like that. So there are actually a good number of opportunities, but I think I might not have utilized the ones in my time, because I realized pretty late like that might be a path that I would be taking eventually. But it's always good to keep a lookout for what's happening. If the employer, like there are employment like workshops like that and things like that happening.
And how did you make connection to the alumnus? I mean, is it just very detailed? How, did you like go to the student council or something and then find out who graduated from? I mean, how did that work?
So I think it's a mix of two different approaches. Some I had known personally, maybe like when I was a first-year student, they were either defending or in their final year. So we had some form of connections. But I also reached out to people I did not know at all, maybe through LinkedIn, but I knew that they went to my program, and I had like maybe introduced myself, what work I was currently involved in, what roles I'm interested in, and if they knew someone who might like maybe even just talk to them, like what they had thoughts about my resume and things like that. And most of the time I would definitely hear back from them, who were always willing to help with those things. And even for like some, yeah.
So through LinkedIn, I mean, you almost like did a cold call, right? I mean, they didn't know you, you just know that they graduated from your university, right?
A few, I think I got introduced through like people I knew as well. So I think it was like all of the approaches, like people I knew, people who I got introduced to through someone, and even like maybe just sending messages in like LinkedIn directly, the cold call, that approach as well.
Okay. Very good.
Yeah, thank you, Nuzhat. Very nice presentation. I had just maybe a personal question for you.
Sure.
What made you switch from academia to what was your, I mean, when you started your school, was your plan to stay in academia, like be a professor or something like that?
I think I was very focused on that track because I
Okay.
did try to like even start publishing even from my very first years for that reason. But I think as I approached my third or fourth year, I tried to understand like what are the main benefits that I might get in academia, and
Mhm.
like getting through that tenure track process seemed more challenging and hard at that point when I thought about like all the struggles of writing grants all the time, getting
Mhm.
rejected, trying to get funds. And I wasn't sure like how much time I would get to do, because I love doing science in the bench or it's in the lab in general. So my point was like, if I was still doing like going through that tenure track, would that actually give me that opportunity? Because it seemed like a big part of that is just writing grants and trying to figure out like to how to make to the next step. But with industry, it's a mix of both actually. Like in my current role, I get 50 to 60% time in the lab and remaining time working on like generating or developing more like studies for the future runs and things like that. So it didn't seem like with industry I won't get that opportunity that I would be getting in academia. So
Right.
also,
Okay. So
yeah,
you're getting a mix of both then.
yeah. And
Mm.
also like it would be also a few more years of postdoctoral experience. Even I actually probably got to like apply for positions like the tenure track professors and things like that.
Right.
But that opportunity is not gone. If you really want to go back, you can always go back, and you would be valued much more because you now have
Hands-on
industry experience, right?
Exactly.
Yeah.
What do you
And
say?
I also, I think I have a much more different kind of experience now working with like a larger number of people as well, because in
Mhm,
my previous experience, I was working in a lab. So we had like a handful of people. We were working in
mhm,
collaborations with other teams, but now it's much different. We work in much larger teams with more people, more opportunity, like even discuss things like teaching experience and all of that. Like I still get a good amount of like teaching experience now because I do train scientists, like junior scientists on a number of even like a number of studies and things like that. So
mhm.
it's a similar kind of condition, I would say, just with much more better funding compared to academia.
Yeah, that's true.
Very nice.
At least you're
Very
enjoying
good.
what you're doing. That's good, right?
Yeah.
That's the most important thing.
Yeah.
Yep.
Thank you, Nuza. That was a very, very nice presentation and
Thanks.
very interesting subject, I would say. I work in the mechanical field. It's much more interesting than that.
And for me, I mean, I am finding very good, similarities in, very nuts and bolts and, this type of industry, same thing. We
Yeah.
try to make our production batches very consistent, right? One
Yeah.
to another. And,
Yep.
the techniques you are applying, controlling such nano objects, people like us should be really reaching out to people like you to understand, okay, now how are you doing it? And can we learn something from that, high technology field to apply to these low tech areas? So again, that I'll keep that in my head, to reach out to you. So again, it was very, very interesting. And
One of the difficulties would be the size of production.
Mhm.
To control the variable in a large scale, it's always a difficulties.
Yeah, I think we have that same thing as well, because when we make it in a lab, it's in like in a 2-liter scale. But when we are doing it in a large scale, GMP manufacturing, it ends up being like a 200-liter scale.
Yep.
So we always have that kind of back and forth running during tech transfer that we have to then figure out like what could be the parameters or like the manufacturing ranges and things like that. So we do that as well, like defining all of those manufacturing conditions. So I guess it's like similar kind of thing as like your fields as well, like where you have to have a very defined process with how like wide that range is for manufacturing as well.
Right. You do the prototype and then you transfer that to production, right?
Yeah.
Yep. Then a whole new ball game starts.
Yeah.
Yep. Exactly. So any other questions from anybody? Sounds like that will be it.
Thank you, Nuzat, again. That's
Sure.
very, very enlightening. And we can publish your presentation in our webpage.
Sure.
Mhm. Yep.
Thank you for also inviting me for this.
Lovely fall morning. You spend it with us. We appreciate that.
Yeah.
Okay. All
And
right.
also, the future Abhiyan who would like to explore the field, I think they now have a number and face to reach out
Sure.
Mhm.
and take the advice.
Mhm. No, it's just really amazing. I know a lot of, my wife is a chemical engineer. Now she's working in softwares, right? Her friend is a chemical engineer working in semiconductors, right? And you with a chemical engineering background working in the biotech. I mean, it's really interesting and amazing.
Yes, so my daughter is studying biochemistry, and I always tell her, you can work in the cosmetics industry with money.
Yeah.
Still,
And get free cosmetics, right?
yes, the ulterior motive, and she always laughed. Thank you, Nuzat.
Thank you.
Did we lost Ashik Bhai? I think so. For some reason, he dropped off.
Yeah.
Yeah, thank you.
Thank you.
Sure.
Nuzat. Yeah.
Okay, I'll log off as well. Thank you.
Enjoy your rest of the day.
Thanks.
Yes.
Bye.
Bye.